Okay. So thank you very much for the invitation, for the organizers, and thank you for participating in this important meeting for many reasons. Now, I'm going to talk about Deuteronomics, which I'm sure not many of you heard yet, but it's definitely coming. It's alive. And the question I'd like to clarify is where do we go from here, being the technology introduced? I'm a retired professor of pediatrics. I was for 20 some years at the UCR School of Medicine, and I would like to thank the European Union consortia for support of this meeting. So Deuteronomics is, just in a nutshell, is how deuterium is distributed in nature and in living organisms, and more importantly, how and why deuterium behaves in nature and living organism in particular ways. And this is, I think, the most important mission of Deuteronomics, is to clarify the role of this particular isotope in in biology, and what is deuterium's role in adaptive biology, disease, and health. For example, oncoisotope was a long time narrative now that is becoming, from the regulatory point of view, cell cycle regulation, a very important topic. Now, you wouldn't be surprised just naming the first introduction of this hydrogen isotope by its mass. And this is the original paper by Dr. Harold Urey, who actually discovered deuterium, and he introduced it by its increase of mass scale compared to hydrogen, which is, we all know, it's 1 Dalton. And I think this is the most, or one of the most important characteristics of this stable isotope of hydrogen, as hydrogen being so active in biology and thrown twice as heavy, twice as large as far as its nucleus is concerned, it definitely makes a big change. For example, just like in a football game, if you throw in a medicine ball, probably the game would stop. So this is something very similar in some biological relations that deuterium is carrying in nature and in biology. Now, this paper earned Dr. Urey the Nobel Prize in 1934, and you can go and see the explanation of why this was so important for his discovery of heavy hydrogen. And to appreciate the role of heavy hydrogen in biology, we have to take a short trip down to the deepest and most in-depth compartment of our cells, and this is a mitochondria, or this is the cell membrane. We can see those double phospholipid layers and the proteins embedded. And as we travel into the depth of our cells, there are these little mitochondria, these little worm-like or worm-shaped organs. And if we go through the outer membrane of the mitochondria, we're going to see this beautiful landscape of these really interesting spinning nanomotors, which are proteins, and you can see them here as they spin around and powered by protons or hydrogens, and they actually reach through the inner membrane of the mitochondria, and they produce ATP, or adenosine triphosphate, for us. Now, surprising as it is, protons or hydrogens are the powering atoms of this, or the gradient across the membrane that powers these nanomotors, and some spin some incredible speed at 100,000 rotations per minute. So we are talking about way beyond like Formula One race cars, or like engineers can, and these are actually 5 nanometers in scale as far as the radius is concerned, this spinning part. And there are proteins that are carrying these protons into the membrane, into the mitochondrial matrix, and there is water formed with the help of oxygen using these protons. So metabolic water formation or matrix water formation through these single protons and nascent oxygen in complex 4 is what drives life, or this is what life is about. And here we can see the ATP as it's detaching from the shafts of the nanomotor, but it's actually a very sophisticated and highly hydrogen or proton dependent. So we are actually internal burning hydrogen machines, in the sense that we actually use hydrogens and protons from food to produce metabolic water, and carbon dioxide as the exhaust fume, and ATP as the biological energy we use. Now we close the inner membrane of the mitochondria, and now we can actually travel back to the surface of the cell. Now, where deuterium comes into the picture is best seen in this other animation. I hope you were guys bringing popcorns, because we're gonna watch movies here, at least 1 or 2 more. Now, these are these little nanomotors or complexes of the intermembrane space, and you can see in more detail how the system works. And this is a very sophisticated system in the part that you can see about 250 protons outside. These are protons, so without electrons, hydrogens without electrons. And as you can see, there's a short little tube that brings in 1 proton at a time and attaches to the side of this rotor. And because of electromagnetic forces, this positive charge pushes the rotor in one way, and in the meantime, on the other side, as the proton travels through, actually the proton falls into the matrix, and oxygen is waiting, and it neutralizes the positive charge of the proton, so the gradient remains as it is, and sure enough, there is ATP synthesized, and this is our biological energy, or biological like energy cache, or that's what we can spend on, like all biological processes that require energy. This is the main source of biological energy in the form of adenosine triphosphate. Now, as you can see how fast this machine is running, and when a twice as heavy, twice as large deuteron comes in and attaches to the protein, it actually breaks it. So you can imagine a high velocity spinning device, and all of a sudden it's unbalanced, and there's a static. So there's practically a lot of details of how these nanomotors are destroyed or made less efficient of producing energy by different deuterium concentrations in the matrix. But practically the bottom line is that some of these events in mitochondria, as far as deuterium overload, could compromise biological processes, including energy production. And from then on, we know what's the result of deficient nanomotor function, cellular health, and differentiation and transformation, and so on. Now, the Nobel Prize for these little nanomotors was given to Dr. Paul Boyer in 1997, who was at UCLA. That's my alma mater. I mean, that's my college, that's my university, that's where I did my faculty work, most of it. And the University of California, Los Angeles was a prime or a very prominent place of studying these nanomotors. And the prize motivation was for their elucidation. There are 4 prizes given at that time for elucidation of the enzymatic mechanisms. It's very important, the enzymatic mechanisms underlying the synthesis of adenosine triphosphate. So even though these proteins are spinning at high velocity, we still look at them as enzymes or proteins, very integrated proteins into membrane systems to be able to drive protons into the matrix and form metabolic water, control the process. Now, then Dr. Zuborev's paper came a few years ago, and this was really a eye-opening discovery to many of us. And this is practically just showing how diverse the deuteronomics or deuterium landscape there is in biology. And he was reporting with his team gray seal, for example, deuterium concentrations. And you can see this in his graph up the blue line, this work next to this sampled gray seal. The samples were coming from the rib and from the pelvic bone. And it was interesting that they measured somewhere over 300 parts per million of deuterium content in hydroxyproline, which is amino acids, in bone collagen. And they called it at that time abnormal. Now we know it's more like phenotype driven, but surely this was surprising, but he was not shy of kind of publishing this paper, which is very important and critical to understand what deuteronomics is. This is actually the stepping stone of deuteronomics, just to see and very clearly discussed as not nutritional source, not food source, cannot be reproduced in the laboratory using physics, inorganic chemistry methodologies. And I felt just obliged to attend this meeting and report back on how big change this paper has introduced into the deuteronomics kind of thinking and planning. And we've been doing this for some 15, 20 years together. So this is not new information to me, yet not entirely new. This was very surprising, but apparently data is data, and biochemists and physicists and doctors and physicians, we need to sit down and discuss this further. So this is what we are doing today. These are actually doves in Rome. And what you can see here is a peregrine falcon coming down from the sky at some 400 kilometers per hour. So this bird will break at the last 50 meters, and this is actually a gray shark, which Dr. Zuborev obtained his data chased by the gray seal chased by a shark. I mean, you can tell this is not an easy lifestyle for that matter. And and also killer whales are just going after him pretty often. And this is a kick here. So this is the sample where Dr. Zuborev was taking his rib bone sample, I guess. And this is a swan in the lake of Balaton. So you can actually see how different this lifestyle is. And their deuterium content in their collagen is not any different than what we see in the environment, meaning that over 300 ppm, the water is about 155 ppm where these animals live. So there's no major difference as far as environmental deuterium exposure, yet for some reason, and we now know is phenotype, practically, or living environment and challenges, physical challenges through life. This was actually a very important phenotype-related discussion and data that we started kind of thinking of and explain certain biological phenomena. Now, in the proceedings of the National Academy of Sciences of the United States, in 2024, there was a paper that appeared called Large Enrichments in Fatty Acid Proton Deuteron Proton Ratios Distinguish Respiration from Aerobic Fermentation. And what was really interesting about this paper is that they measured some really huge differences in deuteron and proton ratios, and but in their abstract they concluded that our findings point to the broad potential of lipid proton deuterium-proton ratios as a passive natural tracker of eukaryotic metabolism with applications to distinguish health and disease, complementing studies that rely on complex isotope tracer addition methods. And this was very important because we used to use tracers, glucose, deuterated glucose, and so on to learn about biology. But this paper showed that actually you can do this in the natural deuterium distribution scenario or environment. And there are large, massive changes that you can interpret biologically. So I sent an email to Dr. Zubarev on June 10th, 2024, like a few days after the paper was sent to me by Esther. And so I told him, listen, we would like to emphasize the phenotypic role of this deuterium-proton ratios with adaptive significance based on many of the contributions in the field. And I suggested Dr. Zubarev a few co-authors. And I figured he's not going to reply. He has more important things to do. But I was wrong, because next day he actually wrote back to me and he said, I also think this is very important what he wrote, because this is actually a game changer in the whole field. And I also think that this is what he wrote. I also think that the PNAS paper in question is A, remarkable and a potentially crucial point for paradigm changing, and B, disappointing, as it still sticks to the old paradigm that isotopes are spectators rather than active participants, viewing lipid deuterium-proton ratios as a passive natural tracker of eukaryotic metabolism. So his views went off, apparently, and rightfully. And he continued that it would be great to write a piece promoting the view that heavy isotopes, particularly deuterium, are not merely passive trackers, but are active participants in metabolism, modulating it. If this is the aim of the letter you suggested, I'm willing to participate. Now, this is a very important approach for all of us, simply because his analytical reference studies have shown really challenging deuterium or deuterium distribution scenarios. And I was very happy that he was willing to participate, simply because he's the source of some of this very crucial, very important data. Now, we wrote our letter, which we're going to go through, just covering the major points, just to see how we designed this letter. Dr. Stephanie Sanafis from the Massachusetts Institute of Technology, from the Artificial Computer Laboratories. Two of our co-authors are from Debrecen, from the Nuclear Atomic Research Facility and Institute. I have the links in our slides, and we're going to provide a PDF of this talk so you can do further reading. And Dr. Zubarev took the senior authorship on this paper because of his contributions, and also because the arguments that he provided for the letter, which I'm going to spell out. So we started as the biosynthetic natural depletion of deuterium, especially in palmitic acid, which is a long chain saturated fatty acid, and it's a substrate for mitochondrial proton delivery. When compared to water of fresh growth medium, there is a large change, or there's a pattern that you can differentiate between fermentation, as shown by the authors that I showed. Accelerated cellular growth remarkably decreased deuterium to protium ratios, particularly in glycerol-perspiring cells, by about 125 ppm. Now that's large. If you are in deuteronomics or you are in stable isotope work, if you see differences of 200% or 125 ppm, you start investigating the phenomenon. So the changes were large enough to go beyond of passive natural trackers of eukaryotic metabolism, So we concluded, or the authors of the original PNAS paper that we wrote our letter in response to concluded that it's a passive tracker to complement isotope studies. And we described in more details that the importance of water and product isotope chemistry is evidence, yet we have to change, we have to emphasize, and this is Dr. Zuberev's points in writing, the emphasis from passive to active role of deuterium in the regulation of metabolism in adaptive biology. I went as far as adaptive biology, but Dr. Zuberev provided the arguments of how regulation or regulatory processes will come into the picture. And I truly appreciate his contributions. So we continued as deuterium depletion. Now it's depleted. So we actually changed the rhetoric or the vocabulary of science or deuterium-related science. So it's more clear and more to the point. So actually, the papers that Dr. Zuberev provided showed abnormally high deuterium content. So we went to the point that, okay, so what you found in this PNAS paper that we responded to is not the only and not the first and not the most unusual finding in the sense that, and Dr. Zuberev reported very high or very significant changes in bone collagen, particularly in proline, hydroxyproline, which really points to some biochemical mechanisms as possible mechanisms for some anomalies, which we know now are phenotype related. And it seems like evolution is really dependent on deuterium-related regulatory processes. And this is a field now that is opening up to submolecular proton tunneling event. These are reaction architectures like these nanomotors that I show you. It's a very dynamic system where deuterium behaves like a ball in a china store, seems like in the moving parts, but it's necessary for the static or the structural elements, especially in bone and in connective tissues. And obviously, these investigations are going to be continued. Now, we argued in this paper that some of the data can be explained using basic or traditional biochemistry. And we did write papers about the pentose cycle and some other intracellular biochemical reactions. Again, I provided some papers here that you can read, whereas others are yet to be clarified. And this is what the main mission of deuteronomics is, to clarify some of those compartmental, inter and intramolecular deuterium levels and differences. Now, the compartmental is very important because the mitochondrial matrix is a cell compartment where water is formed, and the inter and intramolecular deuterium levels are very critical to see how specific biochemical reactions are connected to these regulatory processes. So all these have to be studied under the same umbrella. The mechanism of accumulation of deuterons in specific amino acids, proline and hydroxyproline of bone collagen, high above natural mean oceanic water. Now, we are talking about very high, like almost twice or close to twice as high as the environmental maximum deuterium enrichment. And this is a huge phenomenon to clarify and a challenge, simply because no physical methods that the Zuberev tried with, just squared. You can always go as high as the using methods of physics as the environment to do deuterium enrichment, and there's no high deuterium food source for these bone collagen distributions. That means there are biochemistry involved, and we named those as the phenomenon or high deuterium accumulation in connective and supportive tissues and structural tissues seen in predators with phenotypic adaptation to rapid dives in water, or just like the peregrine falcons slowing down and high frictional forces on the wings, explain why they need increased deuterium or deuterium levels in their bone collagen, simply because to remove a deuterium bond, once it replaces hydrogen, it takes about 10 to 15 times more force. If it's a carbon, oxygen, or nitrogen, it's 8 times to 15 times harder to remove. So it actually provides a very significant structural element especially in bone collagen, where in some cases 20% of the amino acids are proline and hydroxyproline. We know hydroxyproline is formed from proline after synthesis, so it's a post-translational, translational modification in the protein, but it's a very important source of structural elements. And these mitochondrial nanoconfinements are very important to kind of go from no deuterium metabolic water to very highly enriched structural molecules. So we go from, like, I would assume 5 ppm in metabolic water. We know this because we can measure now deuterium content in matrix water, and or in human breath that is always depleted depending on the diet. But practically, it's a very important biomarker or surrogate marker of the scenarios that we are trying, biological and clinical scenarios we are trying to clarify this. So we finished our letter that we suggest that deuteronomics, the study of inherent autonomic hydrogen isotope discrimination processes in nature, should be introduced into translational research. It is important to determine the magnitude of intrinsic kinetic isotope effects, as they are critical in deuterium fractionation, yet often misinterpreted. And the human deuterom should also not be ignored as an active player, as opposed to passive tracker in forming the biological reaction coordinate. That's Dr. Zuber's writing, and he provided literature with that, and even slight deuterium enrichment in water acts as an antioxidant. We discussed this, and we will discuss this further. But anyhow, this is what deuteronomics in a nutshell. I wanted to introduce this writing because it appeared in PNAS. That is a really very well argued letter for many reasons, and apparently the editors and the authors of the original paper agreed. Now, this is Mr. Kennedy, who is Robert Bobby Jr., Kennedy Jr., who is now in the new Trump administration in the US. He's taking the challenge of becoming the health minister. His confirmation is currently ongoing, so we don't know the results yet, but simply he understands the importance of ketogenic or natural food sources, or because of the chronic disease epidemics in the United States. So on his Make America Healthy Again profile, you can see I did suggest that based on reviews by the Department of Health and Human Services, Department of Education, Agriculture, Department of Agriculture, I suggested a National Deutenomics Institute, NDI, established in 2025, if he is confirmed. Again, no one knows what's going to happen, and I'm not involved in politics, so I really don't know exactly how those things go. I just do my science part and contributions, and I have been, there's a link up there, so you can actually look at all the details of these proposals. And I just want to thank you for your attention. If you have questions, happy to answer those. Thank you.